So in the last two lectures, we discussed this idea that we can talk about cognitive behaviors on three levels: computational level, the algorithmic level, and the implementational level. And the implementational level in particular is concerned with how cognition is implemented in the brain, right? So what neurons are basically driving the cognitive behavior of interest. And this will come up in this class. And so you need to have, some background, sort of intro psych level background in neuroanatomy and neuroscience. So I'm going to do a basic review of neuroanatomy, neurofiring, and look at a simple reflex in the first part of the lecture. And then in the second part of the lecture, I'm going to look at how neurons encode the intensity and quality of stimuli. This lecture is based on chapter 2 in your textbook. So the central nervous system consists of the brain and the spinal cord. Hopefully everybody remembers this from intro psych. We can further divide the brain up into the hindbrain, the midbrain, and the forebrain, and each of these divisions can be further divided up into various neural structures, right? The hindbrain has the medulla, the pons, and the cerebellum. The midbrain has the superior and inferior colliculi. The forebrain has the thalamus, hypothalamus, hippocampus, basal ganglia, and the cerebral cortex, or outer layer of the brain. More commonly, we actually just refer to the cortex, the outer layer of the brain, which contains the lobes of the brain, versus subcortical structures. So everything below, everything sub to the cortex. So the basal ganglia is an example of a subcortical structure. The cerebellum is another example of a subcortical structure. What you need to know for this course is if we're looking at the cerebral cortex, the outer layer of the brain, how do we divide it up? And so you may remember from intro psych, we can divide the cerebral cortex up into 4 lobes. Frontal lobe in the front, the parietal lobe sort of in the middle, the temporal lobe along the sides, and the occipital lobe at the back. And there are also 2 hemispheres, the left and the right hemisphere. Now all these bumps and ridges are called sulci and gyri. So sulci in particular means divot or valley. Gyri means sort of peak or bump. And there are 2 that are really important. The central sulcus divides the frontal lobe from the parietal lobe. And the lateral fissure here divides the parietal lobe and frontal lobe from the temporal lobe. Now both the hemispheres and the lobes show some amount of functional specialization. So for the purposes of this course, it's important to know, for instance, that the frontal lobe contains primary motor cortex. So that's just in front of the central sulcus here. So when you're moving your hands or you're speaking, there are neurons in primary motor cortex that are firing to drive that movement. The frontal lobe also contains prefrontal cortex, which we'll talk about in this course, in reference to decision-making and working memory. Just behind the central sulcus, we have the parietal lobe, and the parietal lobe has primary somatosensory cortex. Somatosensation refers to the sense of touch. We can also talk about proprioception, where our limbs are in space. This is the sense that allows you to close your eyes and touch your nose. So when you touch your palm, there are neurons in primary somatosensory cortex just behind the central sulcus here that are firing that lead to the perception of that stimulus. The temporal lobe contains primary auditory cortex, so important in perceiving sounds and speech. The occipital lobe contains primary visual cortex, and we'll talk about primary visual cortex a lot in this course because we spend some time talking about visual perception. Down here we can see a what's called a sagittal section of the brain. So just like a side view of the brain, if we were to sort of chop the head in half down through the nose, and you can nicely see the corpus callosum, which joins the two hemispheres, the brainstem here, and the cerebellum. So there are some neuroanatomical terms that you need to know because they will come up in this course, and these are terms that allow us to image the brain and navigate our way around the brain. So for instance, rostral versus caudal. Rostral means in the front, caudal means in the back. Dorsal versus ventral. Dorsal means up above, ventral means below. Superior versus inferior, so above versus below. Posterior versus anterior, in front versus behind. Sorry, the reverse of that, behind versus in front. So why is it important to know these terms? Because in this course, you might come across something in a paper that says there was activity in the left dorsolateral prefrontal cortex. What does that mean? Well, let's break that down. So left means left hemisphere. Dorsolateral, dorsal means at the top. Lateral means near the outside. So another distinction that's not on this figure is medial versus lateral, middle versus outside. Prefrontal cortex, this is in the frontal lobe. So dorsolateral prefrontal cortex means near the top, lateral, near the outside, in the prefrontal cortex. And so we use these terms to describe different parts of the cortex and navigate our way around the brain. Now in this class we'll also talk about neuroimaging, so structural neuroimaging, taking pictures of the brain, and functional neuroimaging, taking movies of brain activity, essentially. And so it's important to know the three ways that we can slice the brain, right? Because the brain is a three-dimensional thing, but pictures are two-dimensional. So we have to pick an axis. And so we can talk about a horizontal section. So that's like looking down on the brain from above. A coronal section, so that's like looking at the brain from the front, or a sagittal section. It's like looking at a side view of the brain. So the image on the left here is actually a sagittal section of the brain. But again, you might come across an MRI image and it'll say, a coronal section of the basal ganglia. What does that mean? It means that you're looking at the basal ganglia, the subcortical structure, from the front, as if you were looking at the person face on. Now the brain, in particular the cerebral cortex, works on the principle of contralateral processing. So what does that mean? Well, it means that sensory signals from the left side of our world tend to end up in the right side of the cerebral cortex, and vice versa. So in intro psych, we talked about this in the context of visual processing. So visual signals from our left visual field end up initially in the right side of the brain, in right primary visual cortex. Visual signals from our left visual field end up initially in the left side of the brain, in left primary visual cortex. When we're talking about motor control, the left side of the brain, generally speaking, controls the right side of the body, and vice versa. So this is what we mean when we say contralateral control or contralateral processing. Now the two hemispheres can communicate via the corpus callosum. So this is a thick band of axons that links the left and the right hemisphere. And this is important because it turns out that some functions, in particular language, tend to be lateralized to the left side of the brain. This isn't always the case, but in most people, I think it's about 90%, language is lateralized to the left hemisphere. How do we know that? Well, we can put somebody in an MRI scanner and, have them do a language task and see, yeah, there's more activity in the left side of the brain. But there's this very unique group of patients known as split brain patients who have had their corpus callosum cut. Why have they had their corpus callosum cut? These patients tend to have very severe epilepsy, and so cutting the corpus callosum actually limits the spread of seizures and reduces the severity of their epilepsy. The problem with cutting the corpus callosum is the two hemispheres can no longer communicate. So visual information from the left visual field that ends up in the right hemisphere can't be passed on to the left hemisphere, where language can be used to speak about it. And so in the video I'm going to show you, you're going to see a very famous split brain patient named Joe, who's being studied by Michael Gazzaniga at Dartmouth College in the US. And Joe is aware of things that he sees in his left visual field. He just can't speak about them because language production tends to be lateralized to his left hemisphere. So I'll show you the video and I'll break it down for you some more. And I've shown this video in Neuroscience, so hopefully you sort of remember how this works. These patients are very rare and very, very interesting.
Okay. But when Joe is given the same task, his two hands operate as if controlled by two separate brains. What's happening is that each half of Joe's brain is given a separate instruction. He's asked to fix his eyes on the cross in the center of the screen. Anything flashed to the right of the cross goes only to his left hemisphere. Things to the left go to his right hemisphere. Because the two don't communicate, each hand does only what its half of the brain sees.
Wow, look at that. It's really like two different people doing the same.
That's right.
Same task.
That's the idea.
Okay, Joe, I want you to keep your hands out in front
In
of
an
you.
experiment that's now a classic in brain research, Mike Gazzaniga over 30 years ago used a similar setup to find out if the two halves of the brain are specialized to do different things.
Ship.
Joe is being flashed a word only to one half of his brain. Words flashed to the right
Storm.
are seen only by his left brain, and Joe can report seeing those words just fine.
Piano. Good.
But when a word is flashed to his right brain,
Didn't see that. Okay. Joe, I'm going to ask you
but now watch what happens.
to draw that with your left hand.
I think I'm lost.
Why don't you try drawing another picture of it over here if that will help you.
All phone.
Oh, okay.
Sure.
It's almost as though somebody has given him a secret communication.
That's
and
right.
now he knows that it's a telephone. But up until then, he was blind to it.
Exactly.
When Gazzaniga first did this experiment, it instantly proved that the ability to speak resides almost exclusively in the left hemisphere. Not until he sees what his right brain is drawing is Joe able to name
He
it.
said church, didn't he? After looking at the picture.
Yeah, but he had to figure it out about as long as we did. That's really interesting. It's a picture here of somebody communicating almost with another person.
And the communication is not occurring inside the head, it's occurring out on the piece of paper.
Yeah.
Blob. I don't know.
You want to draw a little bit more?
So far, Joe has been seeing only one word.
Cat. That's a cat
Things get
sound.
even stranger when he's flashed two words, each to only one half of his brain.
The right hemisphere saw toad. And
And so his left hand draws a toad.
So there's the toad.
Oh, it's a toad.
And this time I was able to guess what was coming.
Will
Now-
we put a little three-legged stool in there later, or what?
Joe's speaking left brain saw a stool. Saying the word lets the hand that's controlled by his right brain in on the secret.
That's great. That's really interesting. And if he had seen that with the corpus callosum intact, he would have drawn a toadstool.
Right.
Not a toad and a stool.
Right, exactly the point. I've been doing this for 35 years. Yeah?
And
It gets me every time.
Yeah, it must.
So as you saw in that video, Joe was able to perceive things, right, in his left visual field. He had some understanding of what he was seeing. He could draw those objects, but he couldn't talk about them. And this is because his corpus callosum is cut. His two hemispheres can't communicate, and the left hemisphere tends to have language. So in the 1800s, a neuroscientist named Raymond Cahol proposed that neurons are specialized cells that allow communication in the central nervous system. In particular, neurons can send and receive information. And so they may receive information from the environment if they're sensory neurons. They may receive information from other neurons if they're interneurons or motor neurons. But the point here of what's called the neuron doctrine is that the central nervous system is made up of these individual things, these individual cells that send and receive information, as opposed to a situation where it's just all one sort of interconnected web. Neurons are discrete things. How do neurons send and receive information? Well, information moves from one end of a neuron to the other, so from the cell body to the terminals, via the action potential. And so the idea here is that the action potential is a rapid change in the concentration of ions across the neural membrane. And I'm not going to get into, the nitty-gritty of the ions and how action potentials work. That actually won't really come up too much in this class. If you want to review that material, the videos for IntroPsych are actually online, and I can direct you to them. This figure kind of breaks it down for you, and it might jog your memory, but the central idea here is that it involves sodium coming in and potassium going out. What's important is that the action potential is all or nothing. So once it happens, once it's triggered, and again, it could be triggered by some sort of sensory event, all right? Press on the palm of your hand, you're causing action potentials in sensory receptors in your skin. Light striking your eyes, it's causing action potentials in the cells in your retina. Once these action potentials are triggered, they happen and they move from one end of the neuron to the other. The size and shape of the action potential never changes. They always have the same size and shape. The only thing that can change is the frequency of neural firing. So information moves from the cell body to the terminals via the action potential. How does information move between neurons? Well, the main messenger, the main transmitter of information are neurotransmitters. So neurotransmitters are chemicals, and when the action potential reaches the terminals of a neuron, it releases neurotransmitters, and these diffuse across the space between neurons, known as the synapse, and these neurotransmitters bind with other neurons on special receptor sites and either make those neurons more likely to fire or less likely to fire. So we often talk about, the presynaptic neuron, the neuron doing the releasing of neurotransmitters, and the postsynaptic neuron, the neuron receiving the neurotransmitters. Now, most behaviors, all behaviors, I would say, rely on what are called neural circuits. So there probably isn't a behavior that's driven by the action of like a single neuron. Neurons interact in neural circuits. And so we can take a very simple neural circuit, a very simple reflex. This is the knee-jerk response that you may have experienced at your doctor's office. And it involves 4 neurons. A sensory neuron. Sensory neurons receive information from the environment. An interneuron. Interneurons allow for communication within the central nervous system. and motor neurons. Motor neurons cause the contraction of muscles. And so in this simple neural circuit, what happens is your doctor taps on your tendon here in your leg with a hammer. This triggers firing of the sensory neuron, which synapses with a motor neuron and an interneuron. The motor neuron causes your extensor muscle in your thigh to contract. The interneuron inhibits the activity of another motor neuron, which causes the flexor muscle in your thigh to relax, and so your leg kicks out. And so the take-home message here is that all behavior, even the simplest behaviors like reflexes, are driven by the action of groups of neurons acting as neural circuits. So how do we get from neural firing to complex cognition? Well, we don't really have, a very clear answer for that question, but maybe we can simplify it, a little bit, and we can simply ask, how do neurons encode the intensity of stimuli and the quality of stimuli, right? So when I talk about intensity, I'm talking about, how bright the lights are, for instance, in the room. How intense is the stimulus? How hard are you pressing on your palm? When I talk about the quality of stimuli, I'm talking about, characteristics of it. If it's light, it's something like color. If it's sound, it's something like pitch. If it's touch, it's something like texture. So how do neurons encode for intensity? Well, as I just mentioned, right, the shape, the size and shape of the action potential can never change. Once an action potential starts, it's an all or nothing event. It occurs. It moves down the length of the neuron. So how do neurons encode changes in intensity? Well, they do that via changes in the rate of neural firing. So if you're pressing on your palm very lightly, the mechanoreceptor in your palm is firing at a low rate and sending those signals to primary somatosensory cortex in your parietal lobe. If you press harder, the rate of firing increases. If you press even harder, the rate of firing increases even more. So the intensity of stimuli in our world is encoded by the rate of neural firing. And that's the same for any sensory system, right? Crank up the lights in your room. You're just basically sending more action potentials per second to primary visual cortex. The quality of stimuli, so its color, its shape, its texture, its pitch, is likely encoded by specialized cells that detect features of stimuli in our world. And so this has been extensively studied in the domain of visual perception. You may recall Hubel and Wiesel's famous experiment, which was discussed in Intro Psych, demonstrating that primary visual cortex has what we call feature detection cells. So in this study, they recorded from primary visual cortex in cats, and they found that there were groups of cells that responded to areas of high contrast in light. So contrast simply means a change from light to dark. Areas of high contrast at specific orientations. So there are cells that responded to essentially a bar of light that were vertically oriented, horizontally oriented, angled. There were other cells that responded to moving bars of light, other cells that responded to very short bars of light. And again, these are known as feature detection cells. And we think it's cells like these that encode the quality of stimuli. And for instance, if we're talking about a different sensory system like audition, we know that there are cells in the cochlea that respond to particular frequencies of sound. So these cells encode pitch, for instance. So I'm going to show you a video of Hubel and Wiesel's famous experiment. And again, the idea here is that they're recording from a group of neurons in primary visual cortex of a cat. And the group of neurons that they're recording from seems to like a bar of light at about a 45-degree angle to the horizontal. Now you're going to hear a popping sound, and that popping sound reflects the firing of neurons. So the more popping, the more the neurons are firing. So in that video, as you could see, the group of neurons they were recording from really liked a bar of light that was angled at about 45 degrees to the horizontal. Again, the idea here is that there are other neurons that respond to other aspects, other qualities of visual stimuli. So how do we know that the quality of things we perceive relates to the firing of feature detection cells? Well, Blakemore and Cooper ran this experiment in the 1970s where they raised cats in an environment that consisted entirely of vertical bars. And so cats were raised in this vertical bar world, and they only developed feature detection cells for vertically oriented bars of light. And they were effectively blind. They couldn't perceive light that was horizontally oriented. And so this study provides evidence that perception is related to neurons that fire for specific qualities of stimuli. So just to summarize this little section, how do we encode the intensity of stimuli in our world and the quality of stimuli in our world? The intensity seems to relate to the frequency of neural firing. The quality of stimuli seems to relate to the firing of specific feature detection cells in the cortex. And somehow, we're not sure how this happens, somehow we go from the firing of these feature detection cells to this complex visual experience that we have every single day. It's a really remarkable sort of process, and we're not 100% sure how we get from the firing of feature detection cells to complex visual perception. How does the brain represent specific complex characteristics of the environment? Right? So if you look at a face or a house or your car, how does the brain represent that perceptual experience? Well, there are three possible hypotheses, three coding schemes as they're called: specificity coding, population coding, and sparse coding. So specificity coding suggests that complex perception relates to the firing of single neurons in your brain. So if you detect, the right features at the right intensity, this leads to the firing of a particular neuron that may make you, let's say, recognize, oh, I'm looking at my mom's face. That is specificity coding. Specificity coding is probably unlikely because it would mean that, for all of the things that we've encountered in our world, for all of the knowledge we know, for all of the different faces we can recognize and different things we can recognize that we see and hear and touch, there are individual neurons that reflect those perceptual experiences. So that's probably unlikely. Population coding suggests that perceptual experience, complex perceptual experience, relates to the firing of populations of neurons. Population coding is probably unlikely because it's very energy inefficient. We want to conserve energy. Our brains want to conserve energy. And what probably makes the most sense is a coding scheme known as sparse coding, which suggests that complex perception relates to a firing of a small subset of neurons. So if you look at a particular person that you know and you recognize that person, there are a small group of neurons in your brain that basically trigger that perceptual experience. These are hypotheses. Again, we're not 100% sure how we get from the firing of feature detection cells in primary visual cortex to complex visual perception, for instance. And we'll talk a bit more about this later on in the course. So it was a very quick overview of basic neural anatomy and some neuroscience that you'll need to know for this course. And so we'll do the lecture review and quiz. What are the four lobes of the brain and what functions are they associated with? And again, I would recommend that you pause the video, jot down your answer, and then press play again. So what are the four lobes of the brain and what functions are they associated with? We have the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. For the frontal lobe, you should think about motor control, language production, which I actually didn't mention in the lecture, but it occurs in the frontal lobe, and working memory. For the parietal lobe, you should think about somatosensation and attention, which I also didn't mention, but it will come up later on in the course. For the temporal lobe, you should think about audition, language perception, and for the occipital lobe, you should think about vision. What's the neuron doctrine? Question 2, what's the neuron doctrine? So the neuron doctrine simply states that the nervous system is made up of individual cells called neurons that can receive and transmit information. And again, maybe that seems like super obvious, but in the 1800s, there were other hypotheses. Maybe it's just like one big, connected net of cells, like one giant cell, for instance. They weren't sure. And so it was really, when microscopy advanced to a particular point where we could actually see individual neurons, that we realized that the nervous system consists of these individual discrete cells that send and receive information. How does neural firing code for the intensity of perceptual experience? How does neural firing encode for the quality of perceptual experience? So any sensation, we can think about its intensity and its quality. How do neurons encode for these two things? So question 3, how does neural firing encode for the intensity of perceptual experience? How does it encode for the quality of perceptual experience? Intensity reflects the rate of neural firing. Quality likely reflects the activity of feature detection cells within sensory cortices. And from there, somehow our brain builds, this complex world that we experience every day. And we'll talk a bit more about that later on in the course when we discuss vision.